Welcome to a new episode of the Telikilaas biology period. In the last period, we have learned about mitosis and meiosis, which is also a part of the syllabus content, which is entitled Inheritance. In continuation with that, we are going to learn monohybrid inheritance, which I have divided into several parts, and today we are going to deal with the first part of monohybrid inheritance. First of all, we'll go through the syllabus content. By means of learning this lesson, you are supposed to know how to define the terms phenotype, genotype, homozygous, heterozygous, dominant, and recessive. You are supposed to know how to state that two identical homozygous individuals are pure breeding, and heterozygous individual will not be pure breeding. And by means of learning this lesson, you should be familiar in using genetic diagrams to predict the results of monohybrid crosses. And as we learn with the genetic diagrams, we will also learn how to use Punnett squares. When we consider about monohybrid inheritance, the classical experiments in monohybrid inheritance was done by Gregor Mendel. And in conclusion of his experiment, he has brought about several laws in genetics, like the law of dominance, the law of segregation, the law of independent assortments. All these are not in your syllabus. And all those basic laws are still considered as the basic laws of genetics. That is why Gregor Mendel is rightly remembered as the father of genetics. And he has done experiments by using the garden pea plant. The scientific name is Pisum sativum. Whenever you write the binomial nomenclature in grade 9, you have learned that the generic name or the genus name should be starting with a capital letter, and the species name starting with a small letter. So the scientific name or the binomial of garden pea plant is Pisum sativum. So he has done a lot of experiments with this plant, and he was very much interested in Pisum sativum or the garden pea plant, because this particular plant was showing a lot of contrasting features. He was growing these plants in his courtyard, and he was very much interested in these plants because they were showing 7 different contrasting features. These contrasting features are being displayed here. When we consider the flower color, some of them were giving flowers with purple color, others with white color. Then I have separated out purple. The reason you will learn as we proceed with the lesson. Then the position of the flower. Some of the flowers are axillary, others are terminal. The seed color or the color of the cotyledon, yellow and green. The shape of the seed, round and wrinkled. The shape of the pods of the legume, it is inflated or constricted. The pod color, green or yellow. Then the length of the stem, tall, and dwarf, or we say short. So these were the contrasting features shown by Pisum sativum, or the garden pea plant. That is why Gregor Mendel was very much interested in growing these plants. He cross-pollinated them, self-pollinated them, and has brought about a lot of experiments and concluded his experiments by means of various statements which are still considered as the basic laws in genetics. And what do you understand by monohybrid cross? Gregor Mendel has done a lot of experiments. In his initial experiments, he has considered only a single feature. So, genetic cross in which only a single feature with a pair of contrasting traits is considered, they are known as monohybrid cross. And those crosses where he has considered two different features, they are said to be dihybrid cross, which you are not learning in your syllabus. So, just remember that in case of monohybrid cross, he has taken into consideration only a single feature. For example, if he has taken the feature as the flower color, then there are two contrasting traits for that, the purple color as well as white color. So, genetic cross in which only a single feature with a pair of contrasting traits is considered, such type of cross we call it as monohybrid cross. And the inheritance of a single feature with a pair of contrasting traits is referred as monohybrid inheritance. And as we learn with monohybrid inheritance, we should be familiar with various kinds of terms before going into the experiments or the genetic crosses. First of all, phenotype. Whenever you hear phenotype in your genetics class, it refers to the observable features of an organism. So, in your exam, if they are asking you to define phenotype, it can be defined in various ways. Some people define it as physical appearance or external expression of the character, but your syllabus says phenotype is the observable features of an organism. For example, again I am showing the same chart that I have shown earlier. Here we could see the various phenotype. Here the flower color, the purple and the white, they are the phenotype. Likewise, the position of the flower, axial and terminal, they are the phenotype. Yellow and green, those are the phenotype for seed color. Round and wrinkled, the phenotype for seed shape. Inflated and constricted, the phenotype for pod shape. Green and yellow, the phenotype for pod color. Tall and dwarf for the length of the stem. So, whenever you hear phenotype in genetics, it means the observable feature, okay, how the character is being expressed out. So, the observable features of an organism. When we consider the humans also, there are various phenotype that you can see in humans. For example, if you take the eye color, brown eye and blue eye, they are the phenotype. The nature of the hair, curly hair, straight hair, they are the phenotype. The earlobe, some will have free earlobe, others will have attached earlobe. So, free and attached, they are the phenotype. Or in the case of the ability to roll the tongue, some of them are referred as tongue rollers, others are referred as non-tongue roller. So, which is the phenotype. Now coming to genotype. For all this phenotype that we have seen, there will be genotype. Genotype is the genetic makeup of an organism in terms of the alleles present. So, genotype is the genetic makeup of an organism in terms of the alleles present. Other day, the previous episodes, we have learned about the difference between gene and alleles, and we have studied that alleles are alternative versions of a gene. And so genotype is the genetic makeup of an organism in terms of the alleles present. And we have seen that alleles are found at the same position on homologous chromosomes, and we denote these genotypes by means of letters. Usually for we use the first letter of the dominant character. We will see what is dominant character as we proceed. So usually we take the first letter of the dominant character. For example, if you consider the length of the stem, the phenotypes tall and dwarf, and we have written for tall the genotype as capital T or capital T small t, which are the genotypes, the alleles, how the alleles are being found, or we say the genetic makeup of the character. And for dwarf, it is small t and small t. The difference you will learn as we proceed with the lesson. And when we choose these letters, we are not supposed to choose letters which are looking similar in the capital and small letters. So in the case of the color of the flower, purple and white, the dominant character is purple, and if you are using P, then P is looking almost similar in the capital as well as small letter. So that is why you can choose any other letter. When you are choosing letters like this, you are supposed to indicate in your answer, you are supposed to give a key which indicate what letter you have chosen in order to represent the genotype. If the question says about a particular letter, if the question paper contains a particular letter, then you are not supposed to use your own letters. You are supposed to use the letter that is given in the question paper. Otherwise, you are going to lose marks. So, genotype is the genetic makeup of an organism in terms of the alleles present, and they are denoted by letters. And when we have seen the genotype, in certain cases we have represented two capital letters, sometimes we have represented two small letters. So we are introducing the term homozygous. Homo means same, zygous indicates zygote. You know that during fertilization, fusion of male and female gametes takes place. That means the alleles from both the parents are brought together. That is why we say homozygous. That means having two identical alleles of a particular gene. We have seen that alleles are alternative forms, and it can have a dominant form as well as a recessive form. As we proceed, you will know it. So just remember that homozygous, having two identical alleles of a particular gene. These two are identical. These two are identical. So whenever you hear homozygous in your genetics class, you are supposed to represent that character by means of two similar letters, either capital or small letter. So just remember that here homozygous for purple flower, capital B, and homozygous for white flower, small b. So homozygous means having two identical alleles of a particular gene. Now coming to heterozygous. Hetero means different. That means having two different alleles of a particular gene. So here heterozygous for purple flower, capital B, small b. Then you can ask, is there heterozygous for white flower? No. The reason is because of the dominance and the recessiveness, which you will learn as we proceed. So just remember that heterozygous means having two different alleles of a particular gene. And always remember that these two statements are given in your syllabus. You are supposed to know to state that two identical homozygous individuals that breed together will be pure breeding or true breeding. Pure breeding or true breeding means they are able to transfer to the next generation only the same type of alleles. Homozygous means having identical alleles, and they will pass only those alleles to the next generation. They are not having two different alleles. That is why they are said to be pure breeding type or true breeding type. Whereas in case of heterozygous individuals, heterozygous individual will not be pure breeding. Why? Because there are two different alleles. So these individuals will be passing on to the next generation either a dominant allele or a recessive allele. For example, if we are saying capital B and small b, some of the gamete will have capital B, others will have small b, or a dominant allele and a recessive allele. So that is why they are not pure breeding heterozygous individual. Now coming to dominant. They are asking you to define dominant allele. Before that, you are supposed to know what kind of character is a dominant character. Dominant character is the character that is expressed out in heterozygous condition. Heterozygous means having two different alleles. And when they are having two different alleles, only one of that allele will be expressed out, because one of that character is dominant over the other. For example, when purple and white flowers are cross-pollinated, in the next generation you will get only purple, because purple is dominant over white. So here dominant allele means dominant allele is an allele that is expressed if it is present. If there is dominant allele, that will be expressed out, irrespective of the presence of the recessive. The opposite is recessive allele. When capital B and small b are there together, that means the flower will be purple. That means capital B, the allele capital B is being expressed out. The dominant allele is expressed out. So dominant allele is an allele that is expressed if it is present. That is how it is defined in your syllabus. Now coming to recessive. Recessive allele is an allele that is only expressed when there is no dominant allele of the gene present. It will be able to express out only if it is here it should be small b. Okay, so just remember that recessive allele is an allele that is only expressed when there is no dominant allele of the gene present. So all the white flowers will have small b and small b. Okay. Complete dominance. The character is expressed out by the influence of a particular allele. Complete dominance, where the character is expressed out by the influence of a particular allele. Here there are two different alleles, capital B and small b. Capital B and small b are there, and here this allele for the dominant character is completely dominant over this. That is why this allele is expressed out. That is because of complete dominance. The character is expressed out by the influence of a particular allele. Only this allele is influencing the character, not this one. Understood? So such type of dominance, we call it as complete dominance. What about codominance? In case of codominance, neither of the alleles are dominant over the other. Two different alleles are there, but neither of them are dominant over the other. The best example of this is blood group AB. You will study more about codominance as we proceed with the study of monohybrid inheritance. This is the allele for A group. This is the allele for B group. If A was dominant over B, then the blood group should be A. If B is dominant over A, then the blood group should be B. But here it is not A group, it is not B group, but a third different group known as blood group AB because of codominance. Your syllabus deals the codominance and incomplete dominance as same. There is no reference to incomplete dominance, even though there is a slight difference between both of them. Okay. And with regard to these terms that you have learned, you are supposed to refer your past paper, go through the past paper and find out this question in the theory paper of October-November 2017, question number 4B1, where they have asked some questions based on these terms. So try to do that, and by means of that you will get better understanding of the terms that you have studied. And also you are supposed to learn all those definitions that we have come across. And in case of genetic crosses, the genetic crosses should be written by means of a genetic diagram. There are a lot of questions where they are asking you to write the genetic diagram, and the format of the genetic diagram is shown here. Parents, you are supposed to write first parents, then the phenotype, genotype, gametes, fertilization, offspring, genotype, phenotype, if ratio, if they are asking you to find out. And at the right hand corner, you are supposed to write the key, what kind of letters that you are going to use. If the question says about a letter, then don't go for any key. You can use that letter that is given in the question paper. So when they say phenotype, against this you are supposed to fill it. This is like an application form. You are going to fill it. So when you write phenotype, you are supposed to write the observable feature there. For example, purple or white. I will be using the flip chart later in order to show you the crosses. Then genotype, where you are going to write the alleles, whether it is homozygous or heterozygous. Then the fertilization part, where you are joining those alleles together to show the fusion, which can be done by means of drawing lines to show the allele linkage, or you can use another mother known as Punnett square that I will show in the next slide. And by means of the fertilization, you get the genotype of the offspring. Then based on the genotype, you are supposed to conclude the phenotype, and you are supposed to write the ratio, whether they are asking for phenotypic ratio or genotypic ratio. I think your syllabus says more about phenotypic ratio. And in using or writing the genetic diagram, there is another method, especially in the area where fertilization I have mentioned. Instead of drawing the lines to show the fusion of the gametes, you can go for another method known as the usage of Punnett square. So where the alleles are represented like this, and by means of the fusion of these two, and capital A and small a here, capital A and small a, then small a and small a, which shows the Punnett square, which was introduced by the scientist known as Reginald Punnett. And this method also you can follow either the line method, or you can go for the Punnett square method. But if the question says to use a Punnett square, then you are supposed to use a Punnett square. So we will see how to write a genetic diagram. We have already learned the format. I am going to write the format again here, starting with the parent, then the phenotype, then below that the genotype, Then here the gametes. Then here is the area where we are showing the fertilization. Then the offspring with first the genotype. Based on the genotype, the phenotype. And the key also you are supposed to write what kind of letter that you are going to use. We are going to see a genetic cross where the Pisum sativum or the garden pea plants with purple flowers are cross-pollinated with garden pea plants with white flowers. So for purple, we are using capital B, and for white, we are using small b. So they are using pure breeding types, that means homozygous ones. So here we are writing the phenotype, that means the observable feature, purple into white. Genotype, they are pure breeding type or homozygous ones. So capital B crossed with small b. Then the gametes. We have studied about the formation of gametes. Formation of gametes takes place by what kind of division? I hope you remember, meiosis. And what happens during meiosis? The chromosome number is reduced to half. That means the gametes will have only one set of chromosomes with one set of alleles. That means only one of this will be found in a gamete. So some gamete will be with this allele, others with this allele. Likewise in this plant. And then the fertilization process where you are joining them. Don't join with this. Here capital B, small b, capital B, small b, capital B, small b, capital B, small b. Phenotype, this is purple, this is purple, this is purple, purple. So this generation is known as F1 generation. And in the F1 generation, all the plants are of one feature. That means that feature is the dominant one. So we are used to pure breeding types, and the character that is observed in the first generation will be the dominant type. So Mendel has got all the flowers, all the pea plants with purple colors. This increased his curiosity, and he self-pollinated these plants. So in the next generation, to get the second generation, he has self-pollinated these plants. So he has self-pollinated the plants in the first generation, and due to lack of time, I am not writing the format here. So you are supposed to know the parents, the phenotype. Now it is purple into purple. The format should be there, don't forget. Then the genotype, capital B, small b into capital B, small b. Then the gametes, capital B, small b, capital B, small b. And the fertilization takes place. And here capital B, capital B, capital B, small b, capital B, small b, small b, small b. So this will be purple, this will be purple, this will be purple, and this will be white. So they are getting the ratio of the phenotype, the phenotypic ratio 3:1. That means purple 3 of them and white 1 of them. So that is the phenotypic ratio that you are supposed to know, which is known as the monohybrid ratio. And if they are asking the genotypic ratio, that means the ratio between these 3, then capital B is to capital B, small b is to small b, 1:2:1. So this is how you are supposed to write a genetic diagram. As we continue with the same lesson, the monohybrid inheritance in the part 2, again we will come across various genetic diagrams. I hope you followed the lesson and see that you are staying indoors, keeping yourself safe, breaking the chain, and see you in the next episode.